Welcome, and thank you for attending this talk in the Next Generation Wastewater Management Conference, specifically in this case on wastewater surveillance. I'm George Ruck, a PhD student at INRAE Ecotoxicology Lab, and I will be presenting this communication on operational application of invertebrate behavior video tracking for online and real-time wastewater surveillance. During the presentation, I'll be going over the context and the objectives, and then I'll look at the material methods and the results and the conclusions from our study. So, for some context in water treatment, contaminants of emerging concern pose a not so old but ever-present problem whereby more and more chemicals are being identified for their potentially detrimental impact on aquatic ecosystems. And these micropollutants in lakes, rivers, seas are generally poorly understood in their transfer and characterization. And to add to this problem of their abundance, water resources are decreasing in availability whilst the need for them is ever growing. And at the heart of their transfer are the water treatment plants. Current assessment in toxicity is insufficient in this field and typically based on grab sampling methods and limited bioassays. So what the figure presents is in blue an unknown majority of pollutants, in red there are identified pollutants that slip under the radar, and then in orange a few well understood micropollutants that are monitored and treated. And from this graph we can grasp the abundance of the contaminants, that in large treatment plants there's likely to be a huge cocktail mixture present at any one time in the wastewater, and the exact time-dependent identification of every molecule present is a long way off. And this is the context of our bioinnovation that looks to determine CEC presence and micropollutant presence in wastewater. And importantly, our effect-based approach is made to be operational for wastewater assessment in real time. In order to quantify the excess in micropollutant presence, I measure a well-known ecotoxicological biomarker, which is avoidance behavior. It's a well-known marker in research, but it's certainly understudied. The benefits of the biomarker being the sensitivity to non-lethal concentrations and the immediate response potential. But I would say that it's only recently become easily exploited with advances in video tracking and technology and software. So the main challenge is then to use behavior and quantify it in a reproducible way for the experiments to measure micropollutant presence. So the lower figure is in fact the video tracking software that I use, showing the difference between an organism in avoidance and in minimal activity. When talking about the sensitivity of such a biomarker, what makes the reproducibility and comparison of experiments difficult are external confusion factors that affect the behavior. So the first objective is in-lab testing of CEC exposure. And to do this, I look to define a procedure whereby the three selected organisms, the Gammarus, Erpodella, and Radix, would reach a state of minimal activity before exposure. And this was a choice that was made to eliminate the confusion factors, conductivity, pH, day-night cycles. And once the procedure was clear, I tested numerous micropollutants to characterize the signals and channel their signal to micropollutant overloads. The second objective is then to adapt the procedure so that it can be operational for industrial use and to validate the procedure through exposure in water treatment plants. So to achieve these objectives, the piece of material that needs to be understood and is essential to the whole process is the ToxMate. So this is Viewpoint's new toy, a video tracking unit that has since been adapted for both lab use and water treatment plant use. It's a compact chamber that we can see on the top right that is connected to a water source and has 3 observation panels, so water constantly running through, one panel for each invertebrate species, each panel having 16 isolated observation chambers for a population observation. And the center of gravity is measured between 10 and 30 times a second to give an output data that can easily be used to calculate the trajectory of every organism. And here's what the ToxMate sees when the Apobdella species is in minimal activity against avoidance behavior. I mentioned earlier that the ToxMate had been adapted to treatment plants, which is what helps add the operational aspect to my study. And Viewpoints, the partners in the project, currently have lots of ToxMate stations set up, mainly in France, which provides me with behavioral tracking data in wastewater effluents over year-long periods. So the water outlet from the treatment plant is bypassed into the ToxMate station, where the organisms are tracked permanently. And the dashboard in the top right just shows one of our partner sites and the dashboard that they see for each of the invertebrates over on a daily basis. So this brings me to the results. And we have 16 curves for each species, giving position, displacement, and speed over the observation period. So for each species, I have a distribution of movements over this period for every experiment. And the introduction of the pollutant ended up being defined to be carried out over 3 to 4 days into the observation in order to reach the minimal activity state. And the idea of getting to the minimal activity state is that it channels the signal to a higher resolution in changes in avoidance behavior, as the avoidance behavior is characterized by erratic movement and high displacement trying to escape from the observation chamber. And so the conditioning period in the laboratory was also defined to be 3 weeks prior to the entry to the ToxMate station, which was what proved effective in diminishing the sensitivity to macropollution, such as changes in pH or conductivity. So taking a slightly closer look at the signal processing aspect, the minimal activity is shown to be reached before the introduction of the chemical. So in this specific case, it's a control test. So it's a spike of no pollutant. And what we can see is that if we look at the lower quantile of movements at every instant, so the movements at the lower end of the distribution, we actually have a very stable curve in red that's close to zero, which is what helped me develop a protocol that was reproducible and could be compared to other results. So now if we take a zoom into the observation period around the spike of the contaminant for a copper spike, typically a heavy metal that is often found in water treatment plants, we can see that that signal is really focused on the micropollution presence when we look at that lower end of the distribution, that red signal. And it's really an instantaneous response. Within less than a minute, we can see the spike in avoidance behavior and displacement of the organisms. So the procedure was defined and then followed for ecologically relevant concentrations of CECs listed to be of particular concern in water treatment plants across France. And this is a subset of the responses for certain contaminants. So for the Gammaris fossarum, looking at solvents, retardants, and pharmaceuticals here. And the laboratory testing shows the repetibility and the consistent nature of the avoidance behavior response to CECs whilst there was no trigger in the macro changes. And we can even start to see differences in the Gammaris response to these different contaminants. And the second objective remains the application of this surveillance in the context of wastewater. So finally, the first thing I looked to do was simply study long observation periods. And data has been accumulated on treatment plant sites across Europe. And the site that I'm presenting here is for a 6-month observation in Lyon, which is a conglomeration that our laboratory is based at. And the ToxMate has been used to characterize wastewater signals over 6-month, year-long periods with no breaks, data points every second. And I'm looking for naturally occurring spikes in avoidance behavior, as the temporal nature of CEC transfer is known to be highly variable. And in the 6-month period shown below, a zoom on one point shows a very consistent response across the 3 organisms in the time. So showing that, validating in some sense that these peaks do occur. And what's interesting is the difference also between the sensitivity of the 3 species, and perhaps how these differences could be used to define signatures in the behaviors. And what is then left to be done to confirm that these peaks are likely to be attributed to discharge in micropollution is to in fact do an on-site spike in a micropollutant, such as the copper spike shown in the laboratory, which is shown on the figure below, and indeed shows a trigger, especially high trigger for the Gamaris and trigger for all three species, when a continuous feed of copper concentration is put into the bypass of the water treatment outlet. And in conclusion, the concrete findings of the ToxMate project and progress so far are to have defined the protocol necessary for experimenting to assess and quantify toxicity of a certain CEC and continue the experimentation for a growing number of pollutants added to a database, to have made use of the minimal activity model and avoidance biomarker to channel the signal towards surges in pollutant presence, which in turn leads to immediate detection of such a sensitive measure to the presence of the pollutant. And all of this has then been made applicable to industrial use for an effect-based approach to wastewater management. Having the three species adds a dimensionality that is currently being used in further research into behavioral fingerprints, so attributing a certain signature in response to a certain type of contaminant. That's work that's underway. And the outlet for this project is to be able to provide operational alerts in micropollutant presence that could then act as an aid to the decision in the treatment, and of course act as an indicator to the toxicity of the effluent, which could in turn lead to water reuse in industrial and in urban environments, and of course better protection of the aquatic ecosystem. So that concludes my presentation on our wastewater surveillance bioinnovation. If there are any questions, I would be delighted to answer. So thank you for listening to my communication from INAE and Viewpoint on the ToxMate technology.